Hello, everyone. Today, it's my pleasure to present you my work about subthalamic deep brain stimulation, mapping non-motor outcome to structural connections. I'm a postdoctoral fellow at the Brigham and Women's Hospital, and I have no disclosures. So you probably know that subthalamic deep brain stimulation is a treatment for Parkinson disease, and it usually improves motor symptoms quite well. So I mean tremor, body kinesia, and rigidity. But we also know that Parkinson disease consists of non-motor symptoms too. And when it comes to the symptoms, deep brain stimulation outcome is usually more valuable. Now, this is very important because we know these symptoms make a strong contribution to the quality of life, especially once the motor symptoms are addressed. So the natural question that comes in mind is, how can we optimize non-motor outcome after surgery? And the solution might be brought by connectomic deep brain stimulation, which is based on the idea that DBS acts by impacting large-scale brain networks. So what I mean by this is that if we take a symptom, so for example, depression, we could identify the networks that are associated with the improvement or worsening of the symptom, and we could adapt our DBS target or adapt the stimulation pattern, the parameters, in order to best engage the beneficial network or to avoid the detrimental network. But the problem we have with this approach with the non-motor symptoms of Parkinson's disease, I would say, is a problem of dimensionality, because there are a lot of symptoms we would like to address. So if we look at this list on the left, even if we focus on the ones that are brain-related, we are still left with cognitive deficits or dementia, apathy, anxiety, depression, so all symptoms that we want to address. And even if we knew all the networks that are associated with these symptoms, it would quickly become difficult to optimize outcome for all of them, as you can see here. So what we did to tackle this issue is that we aim to identify a few main dimensions of non-motor outcome and the associated structural connectivity patterns. In the second step, we aim to predict individual symptom outcome based on these different connections. So to do that, we worked with the amazing and unique dataset from Philip Mosley, which is a dataset of 59 Parkinson's disease patients that underwent deep brain stimulation and that were tested before and after the surgery. So you can see here the electrodes position. And on the right, you can see the list of variables, so assessments we have. So for this cohort, we had assessment of empathy, depression, apathy, anxiety, cognition, impulsivity, and also impulsivity in the shape of impulse control disorders. And if you look at this graph, you will see that at the group level, there was no significant difference before and after surgery in these different assessments. But when you look at the individual patient outcomes, there was a lot of variability, as we expected. So to extract the main dimensions of non-motor outcome in this cohort, we use principal component analysis, and we could retain 4 different principal components. So the first one, you see it here, was related to general non-motor improvement. Why so? Because all the variables loaded positively on it. And we also observed that these principal components correlated with the change in dopaminergic medications, which also goes into the direction of this component being related to a general optimal outcome of the surgery. The second principal component was more specific to improvement of cognition and worsening of trait impulsivity, and also to a lesser extent to improvement of mood. The third one was related the most to improvement of cognition, and the fourth one was related to empathy and worsening of impulse control disorders. In the second step, we aim to identify the fiber tracks that were associated with principal components. And we did that using the approach we call fiber filtering. So the first step is to model the E-fields, which represent the impact of the stimulation on the local neural tissue. And then we use the normative connectomes that are like a very diagram of the connectivity in the human brain. And for each fiber tract in neurostructural connectome, so for each streamline, we would correlate the impact of the stimulation on the fiber with the principal components across patients. Now, at this stage, we use the change in dopaminergic medication as a correlate because we know dopaminergic medication can impact non-motor symptoms a lot. And so this process we repeated for each of the principal components, which allowed us to find the discriminative fiber tract profiles for all of them. Now, how can we predict based on these connections the outcomes? So it's all about the overlap between the E-fields of a patient and the tracts. So you can see here that the overlap between this hypothetical E-field and the yellow tract is quite high, and it's really less for the red tract. So that's how we would determine a prediction of the PC scores. And so then we can use the principal component analysis weights to go back to the space of the clinical variables and to obtain our predicted outcomes. So now we're counting with our results. So we could observe, we could find tracts that were associated with changing along the different principal components, as you can see here. And while these tracts were not always easy to interpret, there was some interesting level of spatial discrimination between them. On the right here, you can see the correlation between overlap with the tracts and principal component scores. Now, what was more interesting is that overlap with these tracts could explain variance in the individual clinical variables, which was our goal. And that's already a success because the PCA solution we used could only explain 67% of the total variance. So far, the issue with these results is that we used the same cohort to build and to test the model. But we could still test for the significance of these predictions using permutation statistics. And what you can see here is that the prediction of these clinical variables was better than chance for 7 out of 10. In the second step, we aim to validate our model on an external unseen cohort, so an independent data set, which was a data set from Berlin with 32 patients also undergoing deep brain stimulation. And so here you can see the electrode positions. So that's what we based our predictions on with the E-fields, of course. And that's how we did things. So to explain a bit, here we try to predict depression in these patients because that's the assessments they have. Depression we know is most represented on the first principal component. So I will focus on that for now. And you see that in the 5 patient, for example, the ETA or the E-field overlap a lot with the red tract, so the tract that correspond to PC1. And in this patient, patient 31, there is almost no overlap between the E-field and PC1 tracts. So that's why this first patient gets a high prediction for a PC1 score, and this one gets a low prediction for a PC1 score. And so based on that, of course, we have prediction for the 3 over scores, over PCA, sorry. And then we can use the PCA weights to go back to the space of the clinical variables, as you see here on the left. And so we get a prediction for each of the variables. But in this cohort, we only have depression, so we can check the prediction only for depression, and that's what we get. We had a significant correlation between predicted and observed depression improvement, which means that we could predict with our protracts depression in these patients. So as a conclusion, we could map changes along an extensive neuropsychiatric curve battery to the modulation of protract bundles in the human brain. And the degree of modulation of these tracts was able to explain variance in individual symptom outcomes. We believe these tracts could be used to predict and improve the outcome of deep brain stimulation in Parkinson's disease patients. To finish, I would just like to have a quick summary of the work we presented with my colleagues during this session. So we started from mapping disease-specific tracts associated with subthalamic deep brain stimulation. So we have tracked for dystonia, Tourette syndrome, Parkinson's disease, and OCD. Then we zoomed in a bit in order to explore symptom-specific tracts, and we did that for OCD. And we also did that for Parkinson's disease, both for the motor and for the non-motor symptoms. And we hope that this work can help tailor DBS to the individual patient symptoms in the future. With that, I would like to thank all my dear coworkers, especially our PI, Dr. Andreas Earl. And I would also like to highlight our Stimulating Brain virtual monthly talk series Please join us. You can find all the information on our website. Thank you very much for your attention.